Hello, everyone. Welcome to Blockchain UBC's December monthly research talk featuring the esteemed Dr. Ibrahim, who is an expert in blockchain technology, data privacy, and cybersecurity with a focus on secure and decentralized systems. He is currently an assistant professor at the School of Computing, University of Fraser Valley. Prior to this, he worked as a postdoctoral researcher at our University of British Columbia. His contributions include leading the CLEO project, funded by Genome BC at Blockchain UBC, where he worked on developing a secure data sharing platform for genomic data. Welcome, Dr. Ibrahim.
Thank you so much, Stephen. It's lovely to be back to Blockchain at UBC and present the talk. So thank you so much for introducing. So I think we can start now. So as Stephen has already introduced me, so I'm working as an AP at University of Fraser Valley. Before that, I was a former researcher at Blockchain at UBC. So today's talk is actually inspired by the work I did in the project that was funded by Genome Canada during my stay at UBC. So today we will be diving into decentralized and private health data sharing with a focus of empowering patients using blockchain technology and privacy enhancing technologies. So as healthcare continues to evolve, so does the way we manage and share sensitive data. In this talk, we will particularly explore how decentralized systems, especially blockchain, can address security challenges by giving patients more control over their data. We'll see how privacy or enhancing technology or PETs can be used alongside blockchain to further enhance patient privacy. So let's get started. So I'll start with a brief outline of today's presentation. So we will start with the concept of learning health system, okay, and its role to enable precision medicine, which is actually a form of personalized medicine. Next, we will discuss the importance of genomic data sharing and the need for a secure data sharing platform. We will highlight the focus group studies which we conducted with actual cancer patients during this project that actually informed the design of a dynamic and informed consent management system. So through these focus group studies, we evaluated some themes and design features, and through that, we actually extracted some requirements, which were then being used to develop a consent management system. So that consent management system, we call it SecureConsent, and I'll be presenting what we have done in SecureConsent, the architecture, and also a screenshot of the user interface. But privacy still is a major challenge. And lastly, we will look at two privacy enhancing technologies. These are actually tools that actually can be used to enhance privacy. You can use the data without actually seeing the raw sensitive information. So we will actually try to compare different privacy enhancing technologies or PETs to see which one actually can be used in secure genomic data sharing to enhance privacy. Then we will wrap up the question answer sessions if you have any questions that I can address to. Okay. So have you ever wondered how health systems can improve themselves constantly without waiting for years of research? So that's exactly what a learning health system or an LHS aims to achieve. Okay. So a LHS is like a smart feedback loop in healthcare. It continuously collects data from patients, analyzes that data to find patterns and insights. Then these insights can be used to improve care and help doctors make better decisions. So let me break it down into simple characteristics of an LHS. So the LHS is data driven. So LHS heavily relies on the collection, analysis, and interpretation of health data. This health data can come from various sources such as health records, clinical trials, or even patient-generated health data from health IoT or smart devices. Then there is continuous learning loop. And LHS keeps learning from new data and refine healthcare practices. Then there is real-time improvement. So unlike traditional methods, LHS allows healthcare providers to make improvements in real time. As new insights emerge, practices and treatments can be adjusted immediately. So that's completely different from what today's widely used healthcare model is. So right now, evidence-based medicine, so which we call EBM in short, so this is the most widely used healthcare model. EBM relies on research such as clinical trials and systematic reviews to create treatment guidelines. However, any changes in healthcare may take several years. It's a very slow and static process. On the other hand, LHS uses real time data from everyday healthcare practices to adapt and improve continuously. So, LHS is also patient centric. It actually incorporates the needs, values, and preferences of patients. That is actually central to the entire process of LHS. So, patient involvement in LHS is crucial in all stages from research time to implementation and then evaluation whether the treatment is suitable for patients, and there is a feedback loop in LHS. So LHS does not seek to replace evidence-based medicine or EBM. Instead, it enhances it by introducing personalized real-time feedback, allowing healthcare system to adapt more quickly and effectively. So in essence, LHS fills in the gaps left by traditional EBM. It will help to ensure that the healthcare system can continuously evolve based on both the scientific evidence from clinical trials and also the practical patient specific insights. So now, there is a very key use case of learning healthcare systems, and that is precision medicine. So, precision medicine personalizes treatment by considering patient unique characteristics. In precision medicine, genomic data plays a vital role to understand an individual's genetic makeup in order to determine how they might respond to various treatments. So this allows the doctors to select the most effective treatment. So it is more personalized and checks each person's genetic data and evaluates which is the appropriate treatment for that particular person. So it's a personalized treatment option. So a great example of precision medicine in action is precision oncology. So, in precision oncology, genomic sequences is conducted for the tumors and cancers to generate insight into cancer characteristics and biology. That will actually allow to predict how well the cancer will respond to the treatment. This leads actually to more accurate cancer treatments, and that's what research has been concluded that using the genetic makeups, we can actually know more about the cancer and advise better for personalized treatments for each individual. So as treatments progresses, a learning healthcare system can then continuously learn from patient outcomes and improve future treatment recommendations. Okay. So that's what was the story. We talked about learning healthcare systems and why it's more patient centric. And then we have a very special use case, which is precision oncology. And we saw that genetic data is very, very important in these scenarios where genetic data is being analyzed. Okay. But the issue is actually precision medicine aims, or precision oncology aims, to provide personalized treatment based on patients' unique genetic makeup. But for this to happen, we need a vast amount of genomic data that can be shared and analyzed across institution, and that's the biggest issue right now. So in precision oncology, in precision medicine, or the precision oncology, genomic data sharing is absolutely crucial for its success. So without data sharing, the full potential of this approach simply cannot be realized. So the genomic data sharing enables healthcare providers to tailor treatments to individual patients. It also accelerates research by allowing scientists to uncover genetic makeups and disease patterns. This will then lead to, new treatments and diagnostic tools. So, in short, genomic data sharing is the backbone for precision medicine. However, genomic data sharing has its own challenges over here. And we need to address these challenges in order to enable genomic data sharing, which at the end would allow precision oncology to be, realized. So one of the major obstacles over here is the data silos right now. Okay. Each hospital has its own data repository. So data is actually often fragmented across different systems, making it very, very difficult to access and share. So additionally, genomic data contains very sensitive information, and patient consent before sharing such data is very, very important. And ensuring the privacy and security is another big challenge which needs to be addressed. While if the patient gives consent, how to actually provide the privacy and how to share in a secure manner. And that's where blockchain comes in. But before that, we can see there's a global alliance for genomic and health, GA4GH, which is actually an international coalition made for responsible sharing of genomic healthcare data. It actually creates a framework which helped us a lot to see how to develop a secure data sharing platform. So it creates a framework to share genomic data inside a country and also like globally. So, uh, so it allows actually sharing of genomic data in a way that benefits society while maintaining all the ethical, privacy, and security standards. So there are few key principles over here that I would like to discuss. Okay, so the first one is actually the respect for individuals. So the principle emphasizes the need to protect individual rights, dignity, and privacy. So it actually underscores the importance of informed consent. Okay, so that will actually ensure. Sure, I'll just provide the link also. So this prints, or you can just Google, you will be able to find the link easily. Okay. So first is the respect for individuals. So this principle emphasizes the need to protect individual rights. So the importance of informed consent is presented where individuals need to be fully aware how their data will be used and have the right to withdraw their consent at any time. Then we saw that there was another key principle, which was transparency, which is also critical to genomic data sharing. And the framework actually advocates for clear communication about the methods used to collect data how the data will be used, and the outcomes expected for that. Okay. And then we have the accountability. So the framework emphasizes that the data sharing practices must comply with the legal standards and ethical guidelines set by, like, different countries have different ones. So GDPR in Europe, US has HIPAA, and Canada has its own. So accountability ensures that the data is being handled in a responsible manner, and the compliance of the regulation has been fully addressed. And that's where blockchain can help you a lot with the accountability issue, because it records everything and it is transparent. But what it cannot help is with the privacy. But privacy again is another key principle. So ensuring patient privacy is actually fundamental to the responsible genomic data sharing. So the framework emphasizes the importance of de-identifying the personal information, which ensures that any shared data cannot be tracked back to the patient. Again, it's also like highlights the robust security measurements for, like, when you access it, protecting against unauthorized access, and safeguarding all the personal information, while researchers can actually use the genomic data for meaningful you know advancements in the treatment or the healthcare. So here were the key principles, and that actually helped us a lot when we were actually designing a system. So to facilitate genomic data sharing for precision oncology, there is a need for secure data sharing platforms that actually adheres to all the key principles which we have actually set before that, and also are actually compliant with the regulations which are being set for genomic data sharing. So the secure data sharing platform, or SDSP, are designed to enable data analysis across different systems and integrate the clinical the trials and also the real world data quickly. But the issue over here is the patient participation in the SDSP. To encourage patient participant in these platforms, it is very essential that STSP aligns with patient values and addresses their concerns. So before we can implement any of the system, it's very, very crucial that their design and governance reflects the need of the patients whom for which the data is actually being collected. So the research question over here is, how can we develop and implement an STSP in a way that acknowledges and responds to patient values, expectation, and concerns to enhance especially the precision oncology research. And that's what exactly we did in our project, which is CLEO. So just giving a brief overview before discussing what we did. So the CLEO project aims to understand the patient preferences and design features which were actually used to implement and can be used to implement an STSP. So it is CLEO is a pan-Canadian collaboration funded by Genome Canada, and it is dedicated to precision medicine, and it brings together like a lot of key partners, researchers, clinicians, academicians, um, like for instance Genome BC, UBC, BC Cancer, and Terry Fox Research Institute. So the focus is actually designing learning healthcare system for cancer care. The second, which was actually I was quite heavily involved, was actually a building a blockchain based prototype for secure data sharing platform. So, and you can see the link for the project over here, and there are lots of publications during the duration. So it started in 2021, so there are lots of publications that you can see in order to see the progress which we have done. So I'll particularly focus on the blockchain based secure data sharing platform. So for that, we actually conducted a pan-Canadian focus group with individuals who had personal history of cancer. So in this study, a focus group with 28 participants, the median age was 59.5 years, and it was actually conducted all across different provinces in Canada. In terms of cancer experience, there were 44 participants that had previously diagnosed and 33 that were currently ongoing treatment. So the cancer types include breast cancer, gynecological cancer, and lung cancer. So the survey, so we conducted the survey in the focus group, and the questions were actually focused on including the data governance, the data access requests, consent management, also consent incentives of sharing the data, and then the user interface design, how it should be that actually make it easy and intuitive for patient and also provides trust in the application. So during the focus group, several participants shared their surprise and concern about the current state of data sharing in Canada. So one particularly remarked over here that I was a little surprised to know that it was so siloed and the information was not easily shared within our own borders. So this highlights a significant gap in how data is managed across Canada. Then there was another participant who shared a more positive perspective that I actually put it over here in the slide. So I didn't realize that data sharing across provinces wasn't happening at this point. Anything to increase better treatment, I'm all for. And I've always said that I want to share my information. It can help others in research in some way, and I don't need enough financial incentive. So this illustrates a willingness for patients to contribute to genomic research and precision oncology, so as long as it benefits other. But there were several concerns related to the privacy, certain features that they wanted to add on that make them more trusted to the application. So in these studies, there were initial impressions reveal actually strong desire for more accessible and cross-provincial genomic data. And patients were eager to help and be involved in the precision oncology research and support them. And there were also certain preferences of the patients that we actually saw that we tried to include in our STSP. So we actually analyzed the focus group transcripts on these like several issues: data governance, data access requests, consent management, user interface, and they developed some themes and then design features that actually led to certain requirements for our SDSP. Okay. And here are a few of the requirements which I have shared over here. Okay, so let's start. So the first requirements is patient-centric access control. So the users or the patients wanted a simple, intuitive interface that is required where patients should have full control over who can access their data. So this includes strong, secure measures like authentication and authorization, ensuring only approved individual can access the data. So it was more towards patient-centric access control that they wanted rather than, the custodian of the data having all the access control. Then related to the data request, so before any granting any access, it is actually related to the informed consent where the request should include who is asking for the data, why they are needing, what's the purpose, what they are planning to achieve, and the ethical approvals that will actually allow them to make an informed decision. So the dynamic and informed consent management system, this is the third requirement where, so for informed consent means where patients can understand the potential risk, benefits, and implications of their choice before agreeing to share. Whereas dynamic consent means that patients should be able to revoke or update their consent at any time. So they first shared the data, and after that, they wanted to revoke or change certain parameters or change certain time limit for the sharing of that data. Then there is data transparency, where an unchangeable history of records needs to be soon. That actually provides accountability also to the system. And then patients needed interactive communication, where they can be able to communicate with the data requesters, ask questions, and receive updates about their data being used. Okay. So here is actually a overview of what we built in this project. We named it SecureConsent. So SecureConsent is a patient-centric, dynamic, informed consent management, and it can be part of an SDSP. So this system gives patient full control over their data, ensuring transparency and accountability using blockchain technology here. So there are four core entities: the data owners, the data custodians, and the data requesters. So data owners are actually the cancer patients whom actually genomic data belongs to and they want to share. Then there are data custodians. These are hospitals or laboratories that collect and secure and also store securely the genomic data. These are the custodians. And then there are data requesters. These are organizations like research institutes or pharmaceutical companies that request access to genomic data. So for secure consent, we actually used a hybrid blockchain architecture combining a private consortium blockchain with a public blockchain. So we implemented smart contracts on the private blockchain that actually handled user registration, authentication, consent management, and the access control. And there was actually quite a lot of effort to find out the particular way to authenticate and then allow the consent management in privacy and secure manner, and then provide access control. For instance, we actually use access tokens to allow that. So the private blockchain, why we use that? Because it offers lower costs and faster transactions. Okay. So we used actually Hyperledger Besu for this purpose because it's Ethereum client, and it's very easy to integrate with the existing Ethereum tools and the Solidity language. So there is a lot of material that we can use, and then it is compatible with public blockchains, and that exactly what we wanted to do. So the public blockchain actually was used to enhance security by offering data immutability. So we actually recorded transaction hashes to it, and also stored and verified the consortium nodes in the private blockchain over the public blockchain, so that they can be verified. So we ensured proper accountability, because again, the private blockchain is managed by a consortium, and if the consortium becomes corrupt, then there is an issue to that they can change the blocks in the blockchain. So that's why we used a hybrid blockchain, where we can ensure the privacy also by maintaining the private blockchain, and then all the accountability, transparency, and the immutability features can also be integrated by actually having a hybrid model over here. So the smart contracts for the public blockchain were compatible to any EVM based. So we can put it onto Ethereum, Avalanche, Phantom, any of the public blockchains. So we use test networks, obviously, because it's a prototype over here, but it's EVM based compatible. It can be linked to any cost effective blockchain, and that has a good speed, but EVM based would be preferable. So with secure consent, patients have full control over their genomic data, including the ability to grant, modify, or revoke access at any time. So we used unique identifiers for genomic data on the blockchain. So the data requesters initiate, the access request, whereas data owners, if given the consent, they actually provide access tokens. All of these are actually encrypted so that only the authorized entities can view. And on the blockchain, it is being recorded to verify, but due to the encryption of all these tokens and access requests, no one will be able to view them except for the authorized party. So the data, genomic data, is actually stored in secure data repositories managed by the data custodians, but the consent is patient-centric, and based on the consent, this genomic data can be accessed by data requesters. So for further, details of the architectures and the smart contracts, and also we did a performance analysis for the public and also the private blockchain, you can refer to my publication over here. This is the link for that. So over here there is an, one of the requirements was to make a user-friendly interface. So we have a prototype over here as well. So in this image, actually, there are different interfaces for different, user types. For instance, the data owner, data publisher, and the data requester. This is for the patients or the data owners. So we have three sections over here: data repository, okay, the data access and consent management, and then the activity log. So the data repository section allows patients to manage their genomic data files. The data is displayed in a clear structured table. There are identifiers for each data. So patient can choose to view or hide their data with simple actions given over here to control who can actually access or who can actually request an access. Okay, if they are publicly be available for the requesters to request and see the identifiers and then request that. Okay, so if the patient has some genomic data files and it doesn't want to actually even display them for requesters to actually request, then it can also hide them. So in the consent management, patient can handle the data access requests. They can see information about the requester, the purpose of data use, ethical improvers, compliance with regulations, everything which actually provide an informed consent over here. So the important thing is that if needed, they can also revoke access, and the access token can actually be modified as well. Then we have the activity log over here, which records and displays all the interactions with the patient data, such as uploads, access requests, grants and denials, and also revocations. So this log keeps patients informed and provides a full history of their data transaction, making the system accountable. So I think we have 20 minutes left. So this is actually the page for the requester, where through this interface, data requesters can upload research projects and ethical approvals, search for anonymized patient data, and use those data identifiers to request genomic data. And then it can be there are features of answering different questions of patients, and over here you can the approved files can be accessed and used in their research. And then there are like also features to upload data usage reports. These were actually all of these features were extracted from the focus group, and we tried to incorporate everything over here, making them functional requirements. And then we have non-functional requirements such as providing the security and privacy, and that's what we focused using the blockchain architecture to provide those things. Okay, but with the robust consent management and the decentralized system, we give a lot of control to patients of when and how and with whom their data is shared. So it provides more trust to the SDSP, but still it is not sufficient to fully protect their privacy. So there are key challenges still remaining that needs to be addressed. So the first one is de-identified genomic data over here. Okay. So this like even when genomic data is de-identified, there is still a risk that it can be linked back to the patients. So in our system, the genomic data was anonymized. There was no identification or personal health information linked to it, but still there can be many informations that can be extracted from the genomic DNA makeup. Okay, so this creates a a challenge in maintaining the patient privacy. So patients may be willing to share their data, but might be hesitant to trust third parties, especially when it comes to genomic information. So it is very important to establish this trust and resolve this privacy issue. Then there is data double spending. Okay. So this is actually a term coined by Wiki, which actually refers to unauthorized use or sharing of personal data beyond what was originally agreed. For example, there was a access request and the patient agreed for a certain time and for a certain purpose that my genomic data can be used for this research. But then it actually is being used for other purposes beyond what the patient originally agreed. For example, genomic data shared for one research project might be reused for another project or for different purpose or even sold to third parties. So there is no, actual technical systems that can prevent, this um, unauthorized use of data after the patient has given the request and the data is being, shared. So it remains a technical challenge to ensure requesters use the data solely for the defined purpose. We can have all the frameworks and all the rules and regulations, but how to technically evaluate or limit the requesters to not use them and to be compliant with all the excess request parameters that they sent. Okay. And another challenge which we saw, particularly from the data requesters' point of view, is that in many cases, they actually need specific identity information, such as patient age or consent type, to ensure that a genomic data is relevant, or even some, electronic health records they need for their research. So, for instance, research focusing on patients under 40 would not require data from older patients. Okay. So, these checks ensure data is properly used in research while maintaining the relevance. So, however, sharing any part of patient identity again increases the risk of privacy breaches. So there needs to be a way where, identity information can be verified, but without revealing actual identity of the patient. So that's where privacy enhancing technologies come. Okay. So the privacy enhancing technologies or PETs are tools and methods designed to protect sensitive data during collection, processing, and sharing. So particularly these technologies allow secure data use and sharing without exposing the private information. Okay. So the pets can broadly be categorized into algorithmic and architectural pets. So the algorithmic pets focus on privacy during the computation process, while the architectural pets focus on data is structured, shared, and processed. It's in the at the architecture level, so that there is privacy is ensured by design in architectural pets. So here are a few examples that are actually state of the art, and lots of research is being done over these, tools and techniques. So some of the major algorithmic pets include homomorphic encryption, differential privacy, and zero knowledge proofs. So in homomorphic encryption, so I'll just give a brief, introduction of each, and then we actually conducted a survey to see how mature these technologies are and whether they actually can be applied to STSP, particular for the precision oncology case, where we use extensive, data sequences for genomic data. So that was our actually purpose, that how mature enough they are and whether they can currently be used in the STSB to ensure the privacy of the genomic data. So that was our target over here. Okay. So the homomorphic encryption, it actually allows computation to be performed on encrypted data without decrypting. So the data remains encrypted and the processing algorithms actually apply on the encrypted data, and then the results are only decrypted by the authorized people, and actually the person who is actually applying the algorithm are not be able to see the raw data, unencrypted data. So for example, a hospital can run analysis on encrypted patient data without ever accessing the original information. Then we have differential privacy, which is the most widely adopted, technology or technique in the healthcare data processing. So this technique is sort of, uh, uh, you know, um, anonymization. So it adds noise to data, making it impossible to identify individuals. So in a group of, data, and when there is a data set, so differential privacy can be actually employed. For instance, a health organization analyzing patient data to identify trends in disease outbreaks without actually identifying a single patient information, but actually the whole data set architecture or the structure remains intact, so that the, it can be processed and the results can be extracted from that data sets without actually identifying any patient. Okay, then we have zero knowledge proofs. These are actually allow you to, these are also, you know, a tool from cryptographies, and it actually allows someone to prove they know a piece of information without revealing the actual data. For example, this can allow users to prove their identity without exposing personal details. Okay, like age, for instance. They can prove, for instance, they are above 18, but they actually do not show their age. So then we have architectural pets. So these are pets by design. They focus on how privacy is maintained at the system and organization levels. There are two major techniques over here: federated learning and the secure multi-party computation. So the federated learning method trains machine learning models across, decentralized location. So only the model updates are shared, not actually the raw data. For example, an health application can improve its predictive features without ever accessing the user data from the hospitals. So your model actually goes to the data repositories, and the updates are actually, model updates are shared, and you can use them to actually create a complete model, and then that can be used. So it's a decentralized machine learning architecture, which is actually very popular these days and widely being used. Then they have multi-party computation, or the secure multi-party computation, or the SMPC. So SMPC is again a privacy preserving technique that allows multiple parties to jointly compute a result with using the private data without actually revealing the individual input to one another. For example, several hospitals could actually analyze the effectiveness of a treatment without sharing sensitive patient data. Each hospital input will be with their anonymized patient outcomes, will be integrated into the SMPC. The system then calculates the overall treatment effectiveness across all the hospitals, but none of the hospital can actually see the raw data from the other hospital. Okay. So we did a comprehensive survey of pets, and especially in healthcare data. And then when we tried to see in genomic data and data sequencing, what are the techniques being used? And there is actually a summary which I would like to present over here. And we tried to compare each pet with actual challenges and the pros and cons, and then we tried to see how they can be used in the SDSP for genomic data sharing. So for the homomorphic encryption, this technology is partially ready for genomic data sharing. It allows secure data analysis without, the need to decrypt the data. However, it is very computationally intensive, and especially with the large genomic data set, it limits the scalability. And it is very, very computationally intensive. While still this is a challenge and there is lots of work being done on homomorphic encryption, I think the advancement in hardware will actually make it more practical. But currently it is not fully suitable for widespread use in the, you know, SDSP for genomic data sharing. So for the differential privacy, this is the most widely adopted PET. It is ready for use in genomic data sharing. It works by adding noise to the data, ensuring privacy while still allowing researchers to drive meaningful insights. Okay. But there is actually a trade-off between, the privacy and the originality of the data in differential privacy. So for personalized treatments, the accuracy of differential privacy can be an issue because the more noise you put, the more privacy you can achieve, but it will actually affect the overall results that are being processed. Then we have zero knowledge proofs, which are emerging technologies and actually are being they are actually being used in layer 2 solutions for blockchain. So we observed that they are a very good tool in demonstrating the certain identity features, verifying those without actually revealing the identity, and they can actually be used for this purpose in the SDSP, where the researchers can verify certain identity features without actually knowing the identity of that patient. So federated learning, this technology is ready and has already been successfully applied to genomic research. So it allows machine learning models to be trained across multiple institutions without sharing raw information. But the multi-party computation over here, the secure multi-party computation, this method is partially ready, but its application is actually limited by the high computational overhead, particularly for the large scale, genomic data sets. So I think we are at the end and I am also about to end. So let me overall conclude my talk over here. So first of all, in conclusion, genomic data sharing is crucial for advancing precision medicine. By enabling researchers and healthcare professionals to access diverse genomic data sets, they will actually be able to develop more targeted and effective treatments. Now, for the secure data sharing platform, blockchain can play a key role in creating patient-centric system. It can be actually be able to enable consent management and access control in a decentralized manner, ensuring data integrity, transparency, accountability throughout the process. And when it comes to privacy protection, there are several privacy enhancing techniques that can be used along blockchain technology to actually enhance the privacy of patient genomic data. So we have differential privacy, which is a mature pet that can actually ensure privacy in an aggregated data. But it comes with a privacy utility trade-off, which may reduce the effectiveness in precision medicine. So, however, federated learning is an architectural pet which can be combined with blockchain technology to provide a strong solution, which like at the end you will have this secure consent where the patients will actually give access, and then those data can be actually be processed within the federated learning environment, so that actual data doesn't leave the data repositories. Okay, and the precision oncology research can go on. Then we also envision that ZKPs, zero knowledge pools, can enable and enhance the self-sovereign identity, and we actually implemented several aspects of self-sovereign identity in our SecureConsent, and it can be particularly used for feature verifications to enhance privacy and security in genomic data sharing. So in the end, we actually leveraged a combination of blockchain technology, federated learning, and some other privacy enhancing technology, for instance, ZKP, to work along to actually provide a secure data sharing platform for genomic data sharing to actually enhance the precision oncology initiative. So a lot of work has been done in the project, and a lot of work needs to be done in order to completely provide an SDSB, and then that can actually be implemented in Canada. So that's actually our future goals to actually enhance the SDSB, especially when it comes to the privacy, and make a prototype for that. So thank you so much. If you want to connect with me, please use my LinkedIn to connect with me. And thanks again for I would like to thank Vicky and also the blockchain at UBC team and Stephen to actually giving me an opportunity to present today my research. So I was pretty excited, and I really enjoyed presenting an overview, and you can see a lot of research challenges within my presentation that everyone can work on. So I think we have few minutes, and I would like if there are few questions that we can answer them. So I have a few questions I see. So yeah, that's the correct link for the GA4GH. So there's another question. So STSP is standard model to follow for the data security over change solution, or is it just rule set or processes that can be customized? So it's not particularly for the blockchain solution. It actually is a framework, okay, that needs to be constructed, that actually incorporates all these standards and the privacy laws that are being incorporated or being set by different ethical and regulatory authorities. And then we actually envision that blockchain technology and privacy enhancing technologies can be combined to make a technical solution that can actually satisfy these requirements from the regulatory authorities. I don't see any other question, but
Thank you, Dr. Ibrahim. Does anyone else have any other questions, or Mir, is there any follow-up with your previous question? I think you're muted, Mir. I think Mir is muted. Mm, let's see if
so Meet, if you have a question, you can unmute.
Well, let's just say that there's no more questions. So thank you, Dr. Ibrahim, for the wonderful presentation, and thank you everyone for attending the December Research Talk, and I hope everyone has a happy holidays. Thank you, Dr.
Yeah, thank
Ibrahim.
you so much, and see you everyone, and happy holidays.
Bye now.